My name is Lindsay Scott. It is September 25th, 2018. I am performing the lab experiment biochemical testing for microbial identification, the methyl red and Vogue's Prosacker catalase testing. So I will begin that experiment right now. Okay, so I have my prepared warmed S. epi and E. coli right here. I let them sit out of the refrigerator for 30 minutes. So they're room temp now. And I also have my original nutrient broths, the E. coli and S. epi. So I'm just going to pair the E. coli together over here. And I have the S. epis together over here. So I'm going to start with the E. coli. I have the nutrient broth that I prepared with my culture days prior. I am just preparing my pipette, kind of shaking it out to dry right now out of the alcohol. And I'm going to take both of my test tubes here. First, I am going to take my broth and sterilize. And I'm going to draw some up into my pipette right here. So again, this is E. coli. So I have that already right here. I'm just going to go ahead and take my MRVP broth for my E. coli, and I'm going to sterilize in the flame. And I'm going to drop 4 drops into here. So 1, 2, 3, 4. And that is my E. coli MRVP broth prepared. I'm just going to set this one aside and I will start with the others. Okay, so now I'm taking my S. epi broth right here in my hand, my nutrient broth. I'm going to sterilize this tube and I'm going to go ahead and grab my pipette and dry it off from the alcohol. And now I'm going to go ahead and draw some S. epi up in this pipette. Okay, so I have that. I'm going to go ahead and sterilize this S. epi again. Place that back over there. And here's my S. epi MRVP broth. Going to remove the cap and just sterilize this right here. And I'm going to do the same thing with the S. epi. I'm going to have 4 drops. 1, 2, 3, 4. Dispose of this one. And I'm going to go ahead and sterilize this one again. And now I'm going to incubate my MRVP broths for 48 hours. I'll check up on them. If they need additional time, I will add an additional 24 hours and then I will proceed with my experiment. Okay, so right here I have the test tubes prepared, 2 E. coli and S. epis from the MR and VP broth that I incubated. So I'm going to prepare to do the methyl red first. So I have the methyl red open and prepared here. I'm going to go ahead and take one of these S. epis and do 7 drops of methyl red. 1, 2, 3, 4, 5, 6, 7. And then this is S. epi. And then the next one for E. coli, I'm going to do the same thing. 1, 2, 3, 4, 5, 6, 7. 7 drops, and we'll see how this one goes after I observe it. So this is where I'm doing the catalase testing. I have my S. epidermidis and E. coli plates. I have my 2 blank microscope slides, and I'm going to prepare them with the inoculation loops, and I'm going to smear one of E. coli on one side and S. epi on the other side, and I will drip the hydrogen peroxide on it and see if it is a negative or positive test result.